Okay, guys, welcome to your first online lecture of this distance learning adventure. I'm going to try to keep things short and sweet, so follow along with your note packet, and then we'll be finished with this particular packet. We'll be moving on next class. So where we left off, genetic traits and disorders. So we're going to talk about different human traits. Starting with the simplest ones, the human traits that are controlled by a single gene, just like with the pea plants where, the flowers were either purple or white, or pod color was either yellow or green. In this particular situation, these genetic traits are either one or the other. They're dominant or recessive. Huntington's disease and dwarfism are both dominant traits, which means one capital letter, and then the other letter could be either capital or lowercase. Either way, that trait is going to show up. So this is where I want you to remember that a trait being dominant does not necessarily mean that it's more common. Dwarfism, as you know, is not the more common trait. It's more common to be regular height, so the recessive trait is actually more common. Albinism and cystic fibrosis are both recessive traits, which means they have to be lowercase, which means that each parent would have to at least be a carrier in order to pass that trait on to their offspring. Multiple alleles. We've already touched on blood type just a little bit, and this chart is actually in the lab that we did when the last time we actually met in person. So the multiple alleles just means that it's still a single gene, but there's more than just dominant and recessive. So in this case, the three alleles for blood type are A, B, and O. Now, what that means is that each person still only has two alleles, but it could be any combination of those three, which means that you're going to get more than just homozygous dominant, homozygous recessive, and heterozygous. You have different genotype options. So in order to be blood type A, you could be either AA or AO. Same thing with B. You could have two Bs or have a B and an O. AB you only get when there's one A allele, one B allele, and O is only shown when there are two O alleles, which means O is actually the recessive allele. A and B are both dominant when they're dominant together. That, of course, is called codominance. We talked about that last unit. So A and B, when they show up together, are going to be A and B together, which means the blood type AB. If you get an O allele in addition to an A or in addition to the A and the B will both be dominant over the O, which means that the actual phenotype or the blood type will be either A or B. Again, type O, recessive trait. It is the most common blood type. About 43% of the U.S. population has type O blood, which means that even though it's recessive, it's more common. Now, the diagram of RBCs, if you remember, it's called red blood cells. What this means, your actual blood type is just a determination of what proteins are on your red blood cells. So if you're type A, you have A proteins. Simple, right? Now, the antibodies are the parts of your blood that are in the plasma that are supposed to find things that are not supposed to be in your system. So antibodies fight off foreign things that are not supposed to be in your body. Well, if you have type A, you're supposed to have type A proteins. The type that you're not supposed to have is type B, which means you're going to have anti-B antibodies. Opposite with type B. If you're type B, then you have B proteins and you have A antibodies in order to recognize that you're not supposed to be type A. Excuse me. Someone who's type AB is going to have both A and B proteins, which means they don't have any antibodies that are going to be on the lookout for bad blood types because they have both of those proteins. And type O doesn't have any proteins. O is not a protein in of itself, okay? It just means that it has a lack of A and B, which means that someone who's type O is going to have A and B antibodies because they don't have any proteins on their red blood cells. This chart, again, it's in your in that lab that we talked about, okay? I already mentioned this. A and B are codominant. O is recessive. So the only way you get a type O is if you have two little O's. Now, because type O does not have any A or B proteins, that means that anybody can receive type O blood. It's what we call a universal donor. Because there are no proteins on the red blood cells, any antibodies, they're not going to be able to identify them. They're going to kind of fly under the radar. They're going to be undetectable by the antibodies. So anyone can receive type O blood, which is why American Red Cross is always looking for type O bloods, because O is the universal donor. AB is what we call the universal recipient or receiver, because they have both A and B proteins, which means they don't have any antibodies. So if they don't have any antibodies for A or B, there's nothing in their blood that can recognize certain blood types as being bad. Okay. Now, if someone were to get a transfusion from a non-compatible blood type, so say there's someone who's type A and receives type B blood. Well, someone who's type A has B antibodies. So just like in the lab that you guys did last week, if you have someone who's type A and you transfuse them with a type B blood type, the person whose body it is, they have the B antibodies, and their B antibodies are going to attack the type B blood, and they actually cause it to clot, or it's going to cause that agglutination reaction, which means the blood gets thicker. It means that the heart is going to have to work harder, and eventually your blood could get so thick it could actually close off your blood vessels. Obviously, that could cause death. I'm going to go over, I will be posting another video for the genetics packet to show you practice problems with the multiple alleles or with blood type. Polygenic traits. Poly, of course, you guys should know, mean many. Genic is referring to the genes. So these are traits that are controlled by more than one gene. If it's a trait that's controlled by more than one gene. So this would be, for an example, skin color, eye color, hair color, height, weight. Everybody is going to have a certain range that their genes are coded by. So, for example, we have skin color here. If it's 3 genes and they're all recessive, little A's, little B's, little C's, you have someone on the very light end of the spectrum. Okay. If you have all capital letters, all dominant, you're going to be at the very dark end of the spectrum. And then you can see this nice little F2 generation here. How would you like to do a trihybrid cross Punnett square? I'm not going to make you do that. But you see that you end up getting a lot of different ranges, a lot of different pigments instead of just one thing or the other. So these traits that are controlled for in humans are traits that there's not just going to be one thing or the other, but there's going to be many different possibilities to inherit from your parents. Disorders due to non-disjunction. So first thing I'm going to do is break down this word, non-disjunction. So first, the word junction refers to coming together. So, for example, I mean, you guys are probably going to be starting to drive soon. When you see a street sign that says JCT, that's short for junction, which means you're coming up on there's going to be 2 roads crossing together, 2 routes crossing together. So junction is coming together. Dis is the opposite of coming together, so that means coming apart. And then the non in the beginning, well, that means that it's not coming apart. And that's literally what this means. As we all know, during meiosis, chromosomes are supposed to separate. The homologous pairs separate during meiosis 1, and then the sister chromatids separate in meiosis 2. Non-disjunction means that at some point, either in meiosis 1 or meiosis 2, that doesn't happen, which means it's going to result in some sex cells, some gametes having too many chromosomes, and some sex cells not having enough. So what we call it a monosomy, when there is only 45 chromosomes total in the human zygote. So we know there's supposed to be 23 pairs, 46 total chromosomes. This means they have 1 less. And the only monosomy that can results in a live birth is Turner syndrome, which some of you had in the karyotypes that you did a couple weeks ago. That is when a female is born with only one X chromosome. Any other monosomy, meaning if any other chromosome is missing, you cannot have a male that's only Y. And any other chromosome that's missing, that fetus or that embryo will go through what we call spontaneous abortion, which means it will not actually, it either will not implant or it'll end in a miscarriage very early on in development. A trisomy, tri meaning 3, that's where you have 3 chromosomes where you're supposed to only have 2. So you have a total of 47. So this would be things like Down syndrome, Edwards syndrome, Patau. Those are the kinds of things that, again, those are more of the things that you looked at when you did the karyotype. There was also Klinefelter syndrome and super male XYY. Any one of those would be results in trisomies, which can result in live births. And hopefully, remember from your genetics problems packet or the genetics disorders worksheet, I gave you a few of those, and hopefully you researched them properly. So, non-disjunction can affect autosomes or sex chromosomes. Like I said, autosomal disorder, that's Down syndrome. That is when there's an extra 21 chromosomes. So instead of a pair of chromosomes at the 21st spot, there's 3 chromosomes at the 21st spot. And all the rest of the chromosomes are exactly the same as they should be. The sex chromosomes, like I said, Klinefelter, that would be male, XXY, has to be male because there's a Y chromosome. Turner syndrome is that females have just the X chromosome. Super male syndrome is XYY. And like I said, if a male if there's a zygote with a male that only has that Y chromosome, that embryo will be spontaneously aborted because they don't have enough genes. Without the X chromosome, that embryo will not be viable. So this is what it looks like, whether it happens in meiosis 1 or meiosis 2. So here's meiosis 1. So if this pair of chromosomes right here fails to separate, then it's still going to go on to meiosis 2, but now note that the new cells, there's 3 chromosomes over here and only 2 here, which means the resulting sex gametes means none of the sex cells are going to have the right number of chromosomes. These 2 are going to have 1 too many, and these 2 are going to have 1 too few. Now, if meiosis 2 happens if non-disjunction happens during meiosis 2, then this cell is totally fine, and that's going to separate just the way it should be, and there's a correct number of gametes or correct number of chromosomes in those gametes. These 2 are going to be the ones that are affected. Again, this one having too many, this one having too few. But this is a large part of the reason why females go through menopause, because as you should know already, there is a large portion of time between meiosis 1 and meiosis 2, decades long, which means that the cell has been kind of in an arrested state for that many years, which means that it's more likely to have a problem like non-disjunction. Now, detecting these genetic disorders, they have prenatal tests, meaning before birth. Genetic counselors, they're counselors that are specifically, they're specifically trained to look at chromosomes and genes of a baby's makeup, so they can do genetic screening and test for something like Down syndrome or Turner syndrome or Patau or any one of those things. Now, the most common test is the ultrasound. The ultrasound is completely non-invasive. Basically, if a woman is pregnant and she's seeing a Western medicine doctor, like if she's not doing, like if she doesn't have like a midwife or even then sometimes, but pretty much the majority of pregnancies will have several ultrasounds throughout the course of the pregnancy because it's just sound waves. Nothing invasive. They can put sound waves. The sound waves bounce off the fetus and you can get a picture of what's going on. You can see the heartbeat. You can see the organs and see if there's anything developing developing not as it should be. Amniocentesis and chorionic villi sampling are very similar to each other. Chorionic villi sampling has to happen early in the in development. So between like 6 and 8 weeks, a small amount of placental tissue called the chorionic villi is taken out of the mother to screen for the birth defects. Again, that's a good way. You're essentially getting the baby's DNA. Same thing with the amniocentesis, except with the amniocentesis, they take a needle and they extract a small amount of the amniotic fluid. So you should know that a fetus is developing in an amniotic sac that's full of amniotic fluid. By taking the amniotic fluid, they can get fetal DNA. They can look for genetic abnormalities. Again, what they're going to do most of the time is do this, what you guys already did, a karyotype construction. So putting all the chromosomes in order and seeing if there's any extra, if there's any missing, if there's a piece missing, and so on and so forth. PKU testing is something that's done in the hospital after birth. Every baby in the hospital will be tested for PKU. It's called phenylketonuria. It's a genetic disorder that results in the person being unable to make phenylalanine, which is a vital, it's an amino acid, okay? So if you can't use it, then it builds up in your body and it actually can cause major problems and developmental delays. Now, phenylalanine, if someone is tested positive for PKU, all they would do is they would have to have a phenylalanine-free diet. So the next time you have like a pack of gum or a candy bar or a soda or something like that, somewhere on the label, look for it says phenylketonurics, and it will actually sometimes say, like if it says that, it will say contains phenylalanine. Because if that baby is put on a phenylalanine-free diet and remains on that for their entire life, that's the only genetic disorder where you can completely circumvent any of the symptoms. So they can have a completely normal life. They just have to have a very specific diet. Please see the other video that I'm going to post about how to do those multiple allele problems. And once your packet is complete, make sure you complete the short quiz based on this. It's not a difficult quiz. It's just to make sure that you were listening, paying attention, so on and so forth. Those quizzes after lectures are going to be your participation grade for the remainder of however long we have to do this. I hope you guys are all well, and you'll hear me again soon.